what we found here is that there are many proteins, in fact, 674 proteins, which are enriched in a condition where there is UV exposure. So this is a graphical representation here. You have three conditions without any UV exposure. Here, three conditions with UV exposure. So as I explained earlier, when you expose UV, it activates the diazine group and it cross-link with nearby proteins. And if you don't expose UV, there will be no cross-linking. So you can see the intensity of the UV cross-linked proteins are higher because there is more cross-linking. And also, it is proteomics, and we showed that okay, these proteins are interacting with the lipids and they get increased in presence of UV. We also did some analysis where we used Western blot analysis. It's a way of identifying proteins from a mixture. And we showed that yes, indeed, many of these proteins are enriched in the UV exposed condition. So now we have a list of protein, 674 proteins in the hand, and they are all interacting with the choline head group or choline lipid. So we use some bioinformatic analysis and see what are these proteins. So we found that these proteins are of different enzyme classes, having oxidorestases, transferases, etc., etc. And we used a database called David. In that thing, we found that okay, these proteins are involved in different diseases, and also 250 of these hits were part of a drug bank. So it's the first time we showed that like PC lipids or PC head group is just it's not just a structural lipid. It has interaction with a large number of proteins involved in a lot of biological processes. And to summarize this part, so we found that like choline lipid biosynthetic machinery is highly promiscuous. It can take up to different head groups. We found 674 high confidence protein hits interacting with the choline, and majority of the protein hits were present in cytoplasm and the nucleus. 67% of the protein hits are associated with the diseases. We published this work in ACS Chemical Biology, and a lot of people are working on this metabolites now. So the idea is that people initially thought PC are just standalone lipids on the membrane. They don't do much functions. They are everywhere, especially on they're part of many proteins, interacting with many proteins. So after our paper, people are interested. Okay, fine. They show this many proteins interacting with the choline lipids. There can be many biological function of choline lipids other than just being structural. And there are many researchers working on this, and our paper is getting cited well, and we are happy for that. So this is a work I did in ISER Pune with Dr. Jeet Kalia. If you have any doubts, please ask. It'll be nice because I'll be talking about a different short project after this. Okay. Okay, no questions?
If anyone has any doubt, you can ask questions. Any questions?
What was the purpose of your work?
Yeah, the idea is that in the plasma membrane or in general in cells, there are a lot of lipids. And the phospholipids, PC, phosphatidylcholine lipids, are the most numerous, and you see the maximum percentage. Initially, people thought these lipids are just a structural lipid. There is no function. So as I showed you earlier, in the plasma membrane, these lipids are there on the plasma membrane, and it is protecting the cells from outside. So this is the first time we wanted to show that is there a function for these lipids, and that's why we made this method where you make PC lipids reactive and identify those proteins which are present close to PC lipid, which are modulating cell function. Yeah, that's the main reason we started this project, and yeah, that's it.
And could you go for further application of it?
Yeah, actually, I didn't show much. It's too much biology. I don't want to bore you guys. That's why I use. So we used the top, which is the P32, we did a few interactions, like people shown that this p32 protein interacts with the cell surface and involved in cancer. So what we did, we took cell surface proteins and try to see the interaction of this top hit p32 protein. And in presence of phosphocholine, which is the intermediate of choline metabolism, the interaction is getting perturbed. So we can say that phosphocholines might be involved in the interaction of p32 with the cell surface, which might be involved in cancer progression. But there is no direct correlation from my study. It's like we are opening a group of data to people directly on a disease which I am working on. Okay,
Okay.
can I go to the next round? It's a little different, so that's what I stopped now. Okay, I'll go to the next part. Here, okay. The second project is I'm trying to give some summaries of this thing. If I explain everything, it will be too difficult to understand. That's what. So second part, it's involved in membrane-associated proteins. What are these membrane-associated proteins? So if you remember, the first membrane-associated proteins or peripheral membrane proteins, they don't pass through the membrane. They interact briefly with the membrane, the outer leaflet, right? So you see four kinds of proteins here, and they're just interacting. So if you look closely, this kind of protein, they interact with the membrane with the amphipathic helix. So this protein helix goes and interacts with the membrane, and they interact well, and it can stay there. These kinds of protein, there are interaction is through hydrophobic loops. So this loop structure will have hydrophobic amino acids. So since hydrophobic amino acids can interact with the membrane and it stays there. In some cases, there are GPI anchored protein, a specific kind of anchoring. And in some proteins, there are fatty acids attached to it. In Thomas Bookadale's lab, he's a biologist. He's interested in studying the function of proteins and lipid interactions. So we were thinking about how to capture these interactions. So again, we thought I told him, okay, fine, we can think about something like photocopy. We did synthesized a lipid. So this is a bifunctional lipid. So it's very simple. It was a biology lab. What we did, we took a phospholipid. This is P lipid, phosphatidylethynylamine. So if you see the head group, it's an ethanolamine. Fluorophore, it's called body pie. So this fluorophore, the specialty is that if you excite with light of the particular wavelength, it can emit fluorescence. So this is a fluorescent lipid. And what we did, we treated this lipid with SDA. So if you look into SDA, it has a diazirin, which is a photoactivable compound, and there is uh, NHS group. So if you mix these two things in slightly basic condition, and you get this combination, it forms this NHS reacts with amine, gives an amide linkage, will give you this molecule. It's a very straightforward reaction, and nothing much, no columns. We separated using TLC, and we characterized it by HRMS, and we showed that okay, this molecule can be made. So the one difference you have to remember, this is a lipid. It's not a head group, right? It's a whole lipid. And if you put this into the cells or make liposomes, it lipid bilayer. So once we got this lipid, we wanted to see can be used to capture the interaction of maps or proteins. So what we did, we strategized a technology called PlyMap. As we named it PlyMap because it's a bifunctional protein. It's a method. And we used liposomes. So liposomes, you take lipids and you mix it with them, you dry them and you add some buffer and you pass through small pores. So when the lipid mixture pass through small pores, it formed this kind of structures where it's a double layered spheres or double layered particles called liposomes. And this is typical in vitro experiments people do when we want to study the lipids. You can control the size of this liposomes by um, you know liposomes by changing the pore size of the filter. So if you use a small pore size filter, it becomes smaller, and if you use a large pore size, and we thought like since with proteins, and if you shine UV, it will physical representation. And so this idea, so you take liposome containing our BDP or cross-linkable lipid. We did to test the methodology. We took a protein called sedum. It's present in a bacteria. So you extracted the protein, liposomes containing different lipids. PC stands for phosphatidylcholine, which is a basic lipid, and we have a lot of PI lipids. Are phosphatidylinositol lipids. This is a phosphatidylinositol. This is the backbone of the lipid to fatty acid chain, phosphate group, and there is a inositol head group. So this phosphatidylinositol. So this is a very special lipid. It's a, inositol is present, and in different positions of this OH can be replaced with a phosphate group. For example, if you in the third position, if there is a phosphate here, it's called PI3P. That is phosphatidylinositol 3-phosphate. And if you have something in the fifth position, it's called PI5P. And if you have two phosphate group, it becomes PI34P2. PIP2. It can have different position, and if it is present in 3, 4, and 5th position, it becomes PIP3. So this is a very complex lipid. People were interested in studying, and they were trying to find out proteins which can specifically detect or bind to these head groups. And at that time, sedum is one of the known interactor of PI4P. That means the phosphate group is present on the fourth position. And we made liposomes, and we wanted just to see, can we replicate what people showed. People showed that sedum protein can interact with PI4P better than the other inositols. So what we did, made liposomes, and with different PI lipids, and you add the protein sedum, shine UV, and we separated the proteins on a page gel. So you see proteins are there, kumasibilin blue stained protein. And when we check the fluorescence, you see it binds better to PA4P. So this is something which is just reproducing what people showed in the literature. The sedum proteins can bind well with PA4P. So from our side, it's a validation of our technology, saying that the PlyMap technology can be used to identify of proteins with lipids. Once we knew this, we thought, okay, it's okay, it's all nice, and we were interested in understanding this technology, making this technology good. What is use? So at that time, we were planning to understand the interactome of cardiolipid. It's another lipid. The specialty of this lipid is that it has 4 fatty acid chain, like 1, 2, 3, 4, connected to glycerol backbone with 2 phosphate. And this is one of the most important lipid present in mitochondria, which is the energy house of the cell. And it constitutes 20% of total lipids in mitochondria. And there are no proteome present exactly for the cardiolipid. So we thought, okay, it's a nice starting point. We have PlyMap technology, which can use to identify, or which can identify specific proteins. And cardiolipin interactome was not known at that time. So what we did, we made this is a strategy. So we may the first strategy is to see whether this PlyMap technology can be used to identify, can we pull down proteins from the mixture. As a positive control, we made liposomes with BDP, and we put proteins, we shine UV, and so you see here, when you shine UV, some proteins will bind to the lipids. There will be proteins which are not bound like this. How to separate them from the mixture? So we used a symbol technology called phase partitioning. So the idea is that we use a detergent called Tritonix 114. The specialty of this detergent is that at 2% of TX114 at 4 degrees centigrade, this is a solution. But if you warm this solution, it phase separates into two layers. And the detergent, which take all the hydrophobic part, and the detergent poor layer will have the non-hydrophobic part. The idea is that if the cross-linking happens, and if the protein binds with the lipids, it becomes more hydrophobic. And when you do this phase separation, you'll have more proteins present in the detergent-rich layer of the tube. So that in that way we can isolate those are present on the which are interacting with the proteins. This is the idea, pull down. So for the real experiment, what we did, we made a liposome which containing the BDP lipid, which is the crosslinker, and we also put cardiolipin. So the whole idea is to understand what are the proteins present in the cell lysate binding to cardiolipin. And we made these liposomes and we put brain cytosol. So we have a lot of proteins in the brain cytosol. And in one sample, we shine UV. In another sample, we do not shine any UV. And after shining this thing, we extracted it with the Triton 114. Then, as I told earlier, this protein needs to be cut into pieces. So we use trypsin to digest these proteins. And we label them. So this is the tricky part. So in one hand, you have a sample which is not UV cross-linked. One sample you have cross-linked. So in one side, we used formaldehyde heavy. We label them separately and mix them together and did proteomics analysis. So this is what we did. So you see the protocol is like you have liposomes with the cardiolipin and cross-linkable thing. You shine UV and and what we found? We found that alpha-synuclein is the most enriched protein in this particular thing. So brain has a lot of proteins, but in our case, the cardiolipin binding, the most bound protein was alpha-synuclein. You guys know that alpha-synuclein is one of the major protein involved in Parkinson disease. It's a neurodegenerative diseases. And in those diseases, this synuclein aggregates and it interacts with neurons and prevents the nerve impulse transmission. So we were very happy to see that cardiolipin can interact with alpha-synuclein, which is an important protein. But the question comes: it's just an error of mass spec. You have two samples and mixing up together, and you're doing proteomics. You might by some error is there. That's why we are getting alpha-synuclein as the major thing. To confirm this thing, we did again the typical analysis of Plime. We made different lipids, liposome containing different lipids, PC, sphingomyelin, phosphorylethanolamine, PA, PGP, everything, whatever we have. And we did the same interaction. We purified nuclei, mixed with the liposomes, and it shine UV, separated the page. And we found that alpha-synuclein showed maximum binding. So it's not just a random chance that alpha-synuclein was present in the protein hits. It's really, it can interact with cardiolipin. So it's very important. So and cardiolipin is present in the mitochondria. So my collaborator is working on how synuclein can interact the mitochondrial function of the cell. Maybe we can talk about synuclein can interact with and so my collaborator is trying to find out how can it bind to mitochondria inside the cell and it modulates its function. So it's going on. In summary, we designed and synthesized a bifunctional lipid probe, and we found that BDP cross-linking is specific to those proteins interacting with the membrane only. And the readout of the thing is fluorescence, and with high sensitivity, and it's very easy to make. Even any biology lab can make this. And also, this probe can be used to separate complexes from the complex mixture, proteins from the complex mixture. That's the summary, and we are happy to say that we published this thing in Traffic and Current Protocol in Protein Science, and also we took a US patent, Indian patent and US patent, and we are in the process of marketing or licensing this patent for different companies. We are not yet there, but we are just trying to find. Okay. This is the end of the second part. If you have any question, please ask. I'll be showing one more thing and finishing it.
You have done only the in vitro studies, right? Have you gone
Yeah, yeah.
for any in vivo studies?
Uh, okay, it's a good question. The presentation, what I'm talking about, only in vitro studies. The work, I did not do much of in vivo in the work I showed last two works. I did not do any in vivo work. But currently, when I'm in now, I'm in France, and we are doing a lot of in vivo work, but not exactly on lipid binding proteins. We have other proteins, and we are using knockout mice and trying to understand the function of the protein. But for this particular project, we did not do any in vivo work. All the works are either in vitro or some are in the cell lines, not in the animal model yet.
Okay.
Okay.
Any more questions?
Okay. Okay.
Okay.
This is the third part. Okay. Very simple, very straightforward. It's not much biology. Here, all are talking about AI now. People are. So we thought, okay, can we do something with the AI? I'm not an expert, but still, I looked and see what are the problems which can be solved by AI. So one of the problem, what we found is that very simple problem. So you look at here. This is lipid droplet. These are lipid droplets. So what are lipid droplets? These are the small organelles, or you can say small particles seen in the cells. The main function of this organelle is to store lipids, especially neutral lipids like cholesterol esters or triacyl glycerols, and they store them in the form of lipid droplets, as the name suggests, lipid and droplets. So they are like lipid containing droplets. And the major location of these lipid droplets are ER. It's a endoplasmic reticulum. It's a organelle inside the cell where they store. So if you look here, there can be different kinds of lipid droplets. For example, in the case of budding yeast, it's a yeast. You see, lipid droplets are like this, round, small rounds. If you go to another cell called mouse embryonic fibroblast, you see lipid droplets are much more smaller, but they are very connected with each other like bundles. This is nucleus. And adipocytes, they are the major major cell which stores lipid. And in our body, wherever there's adipocyte, there will be lipid storage. And you can see there are in the early differentiation, when they are young, you have multiple droplets. But when they become older, after differentiation, they become a single lipid droplet containing cell. So I just want to show that there are a lot of variation in the lipid droplets, but the one thing is common: they are all round, and they contain neutral lipids. So this is the representation. This is how they look like. This is an endoplasmic reticulum. This is a graphical representation again. And you have a single bilayer, which is just going through like this, and you have a lot of lipids inside in the lipid core, which contain, as I said, cholesterol, esters, TG, and some other lipids. And on the surface, there are many proteins. It's not important, but just to show this is how they look like. And there are lipid droplets. So why it is important for me? So the work I'm doing here in France, we are working on macrophages. So these macrophages are the protective cells which go through our body and look for any pathogens. If it sees pathogen, it just catch the pathogen and eat it and digest it. So people have shown that this macrophages also involved in atherosclerosis or heart diseases. So the idea is that in the heart disease, these macrophages will go not in, I mean, everywhere, especially in heart. It eats lipids and it become very fat and just get deposited on the blood vessels. This is the first reason why there is a blood plaques in the heart, which can lead to blocking and lead to heart diseases. So one of our work is on this atherosclerosis, and we were interested in studying how this macrophages go and eat these lipids and become fat. And so one of the major thing to look for is the lipid droplets, because these are the place where we store the lipids inside the macrophages. So we can grow these cells in the lab. So we have mice, and we sacrifice the mice and take the bone marrow, grow these cells in the plates, and you can convert those cells into macrophages. Once they form macrophages, we give them fats to eat. So they happily takes up fat and store this lipid as lipid droplets. So this is a cell. You can see there is a nucleus, and these are lipid droplets, and you just divide into two. You have all the lipid droplets here and the nucleus. So our idea is to see what happens when you do some treatment or when you do some knockout of the gene. What happens to the lipid droplets? And as you see, they are small and they're round, but some are connected with each other. So the major output, what people in the literature, is that the number of lipid droplets or the size of lipid droplets. So if you do some treatment or if you do some knockout, what happens to the lipid droplets? Either they are becoming bigger and they have more lipid storage in the cells, or they are becoming smaller, or they are the number is decreasing or increasing. So all these things needs to we need to analyze it. So it's very difficult to analyze from the images. So what people have is something called this kind of analysis. What people do, people take this image, and you see all these bright spots. These are all fluorescent images. You can see, if you look here, you can AI model to segment or to analyze the lipid droplets what we have. So this is what he did, and it's beautiful. And if you see here, once he did this stardust modeling, and you'll see if you compare this and this, it can clearly differentiate a bunch of lipid droplets into separate rounds. See, now I can say we have this many lipid droplets, and these are all the small, all are different color. They are different mask. I can, using analysis software, I can say how many rounds are there, what is the intensity, what is the size, what are the number and everything. So we used stardust based AI platform to understand or to analyze the lipid droplets. So it's unpublished. We are doing a lot of other experiments to publish, but it's a part of the paper for analyzing lipid droplets. So that's what all about. And I would like, if you have any doubts, please ask me.
You can ask any questions if you have.
Work is only on the growing stage, right? You have not completed
Yeah.
macrophages.